Hello to Science 9 students, and in this video we are going to delve a bit more into the structure of DNA, and I'll try and make it a bit shorter than the previous video. Okay, so in this video we are going to have a quick reminder of the analogy of DNA, the cookbook analogy. We're going to see, we're going to do a bit of a deep dive. So we're going to see a bit more detailed information about what DNA is made of, what are those individual parts that we were looking at in the last video, how does DNA store information, and what is complementary base pairing. So three things we're going to sort of dive into after our analogy. So going back to our analogy, remember that DNA is a big molecule. I mean really, 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 really big as far as molecules go. And a molecule is just made up of bonded atoms. Now we can see in the bottom diagram a bit more clearly what the structure of the DNA is. It forms what's called a double helix, which is kind of like a ladder that's been twisted. So this is the shape of it, and it's made up of these individual sort of the handrails of the ladder, and then these would be the rungs of the ladder here, these individual parts that are going across here, the base pairs as I mentioned earlier. So it is a super long molecule, and it is coiled up into tighter and tighter coils and eventually forms a chromosome. So this is a pair of chromosomes. One chromosome would be something like that line, and the other chromosome would be something like this. So those are the two chromosomes, and we have, remember, we have 23 pairs, which means we have 46 chromosomes, which is the same thing as saying we have 46 DNA molecules. Just a very, very long molecule to make up one chromosome. Okay, and if we go back to our analogy, remember that if a chromosome is like a cookbook, it has many, many recipes in it, and the gene itself is one of those recipes here. So the cookbook is like a DNA strand, and then within the DNA strand we have genes, or within a cookbook we have recipes, and our genes tell us what to do or how to build proteins, just like a recipe tells us how to build food. So because we have 46 chromosomes, you essentially have 46 big cookbooks in the nucleus of every one of your cells. And so many genes within it, just like many recipes within a cookbook. So we saw this in the last video here. So now we're going to zoom in a bit more. So on the left-hand side, we've got this double helix again. But this version over here slightly to the right, this is the kind of the unwound version here. If we unwind the ladder, you get something that looks a little bit like this. Now, if you look at this image on the right-hand side, this image is showing the same information over here. So when we look at this image on the right-hand side, these parts here that have these blue pentagons, these represent this, roughly speaking, these individual parts here that sort of form the backbone or the handrails of the ladder, and these parts here, the A, the C, the T, the G's, these represent these individual parts that make up the rung. Now, if you look over on the right-hand side, what we're looking at here, which is a better color, so this right here would be sort of this portion right here. And so you're going to see that there's these pairs on the left-hand side, or there's these portions here on the left-hand side that might represent 4 of these squares over here, and on the right-hand side we're going to have these other pairs right here. So these other 4 parts would represent, say, these 4 parts, these parts on the right-hand side. And we'll get into some naming of that on the next page here. So actually, before we do that, we're going to watch a quick video, just a portion of it. Now, there's a lot of large words that are being used in this video. You don't need to worry about the terminology. If you can answer the questions on your activity sheet, then you've gained enough. This is just a bit of extra information here.
Now, let's move to a more simplified representation of DNA to discuss the details. We can unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has 3 components: the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine.
Okay, so as it says in the video, the DNA is made up of repeating pieces called nucleotides. So a nucleotide is this area that again is highlighted in this box here on the right hand side. So this is a nucleotide, and you can see the nucleotides have 3 portions to them, as were mentioned in the previous video. So this is the phosphate group, and this is the sugar. So this is the phosphate group, this is the sugar, and together they form the sugar-phosphate backbone. These are the individual portions of the hand railing of the ladder, if you will. And they have them on the right-hand side as well. So this would be, let me just highlight that. These would also be nucleotides. These are 4 nucleotides on the left side, 4 nucleotides on the right side here. And you'll notice that they have these these parts here with the letters on them, the A, the T, the C, and the G. These are our nitrogenous bases. So these parts here, let me just highlight one here. Oops. This part here is a nitrogenous base. So these are the 3 parts. There's a phosphate, a sugar, and a nitrogenous base. When you add those 3 things up together, you have what's known as a nucleotide. And nucleotides always form in these pairs. And if we have these pairs, these are the rungs of the ladder that make up the DNA. Okay, so again, just to repeat, a sugar, that is this complex molecule or complex portion of a molecule that is a pentagon shape. We have a phosphate group, so this is going to be a phosphate with various other atoms attached to it, and the nitrogenous base, which comes in 4 distinct flavors, as we can see here. So we have 4 types of nitrogenous bases. We have adenine, we have thymine, we have cytosine, and we have guanine. Now the good news is we don't need to remember these individual names. So don't need to remember. Hopefully you're happy about that. In terms of what we need to remember, we do need to remember the letters. So need to know. So these parts here, we do need to know the A, the T, the C, and the G. These are the 4 different types of nitrogenous bases. Now, again, you do not need to know these 4 names here. Just the A, T, C, and G is fine. Now, some of you may be looking at this and seeing that as they've drawn this here, they have these individual shapes. So adenine has this shape, thymine has this shape, cytosine and guanine have these individual shapes. So think about that. Think about what you're seeing, if you notice any patterns between those 4. And hopefully you may have realized that they have complementary shapes. So this is a triangle pointing inwards, and this allows for a triangle to point inwards. Cytosine has this shape, this rounded shape on the outside, and guanine allows for a rounded shaped molecule to fit in it. That's not exactly what the molecules look like, but it's a nice way of understanding how these fit together. And so these bases are always going to join up in predictable ways. So whenever you have an A, it's always going to be paired with a T, as we can see in this one here, and in this one here. So if it's A on the left, it'll be T on the right. If it's T on the left, it'll be A on the right. And one of the ways that we can remember this is that apples go in trees. So apples in the trees, A and T are going to go together, and also likewise C and G are also going to pair together. So if we've got a G on the left, we're going to have a C on the right, and if we have a C on the left, we're going to have a G on the right. And this is what is known as complementary base pairing. And there's some very important reasons why we need this. So we need this in terms of how we replicate or how we duplicate DNA. And also we need that for fixing if there's been any damage due to radiation, or if we have damage in the cells, we can identify where that damage is. And in terms of remembering that cars go in garage, or you can kind of see that C is a curly shape and G is a curly shape. Those two go together. A only has straight lines, T only has straight lines, so those two will go together. So if you can either remember apples in the trees and cars in the garage, or just looking at the shape of the letters. So why is that important? Let's take a look at this sequence here. So let's say this is one side of the ladder. So this is the phosphorus sugar backbone. Now, in a sense, DNA, for just existing on its own, if we don't take into account the way it splits, is a bit redundant. So we have these 4 letters that spell out the words. And so if we compare this to English, and so in English we have 26 letters that allow us to put together every single word in the English language. It's based off of these 26 letters. So in DNA, there's only 4 letters. And those are A, T, C, G. And when we put these letters together in combinations, we spell words. Those words produce our genes or our recipes. And if we have a combination of a bunch of recipes together, we have our cookbook, or we have our DNA. Now, because we know how these always pair, we always know that apples are in trees and cars are in garage, well, I can tell you exactly what's going to be on the other side of the ladder. I can tell you what's going to be the other nitrogenous bases because of how these partner together. So we know that apples go in trees, and so if we know there's an apple on the top one here, then there will be a tree on the bottom. Likewise, we're going to see T matches with A matches with T, C matches with G, C matches with G, G matches with C, A, and G. So we're not going to go into too much detail now, but maybe you can think about how if you were going to split your DNA, how you could easily make 2 copies of your recipe book. So this right here is essentially a recipe. You could think of it this way. This is could be a gene or a small portion of a gene. So this is one recipe, but if this gets split in half, like so, well, I know exactly what other parts I would need to make up my other half of the recipe. That's all the detail I'll go into for this video, but we will definitely learn more about that in subsequent years and subsequent lessons. Okay, so just to recap, again, going back to our beginning of this lesson, here. So what is DNA made of? It's made up of these nucleotides. The nucleotides themselves are made up of a phosphate group, a sugar, and a nitrogenous base. So that is what they are made of. In terms of how does it store information, well, it uses a short alphabet of 4 nitrogenous bases. Those are A, T, C, and G. These form the letters that make up the alphabet. It's a small alphabet, but they make up the alphabet that spells out all of our recipes for us. And complementary base pairing is this fact that when you have these 4 different letters, we know that A and T will partner together, the apples are in the trees, and we know that C and G will pair together because cars are in the garage. So that's the lesson for today, and I hope it was informative.